So I would like to welcome everyone to the presentation. I'm very excited to be discussing how we are using our Pathology antibodies to build multiplex panels to make it easier for our customers to get started in their multiplex experiments. For those who do not know about Bethel Laboratories, we are an antibody manufacturer located uh, in Texas, and we actually manufacture and validate a range of products, including rabbit recombinant monoclonal antibodies and polyclonal antibodies. And we have the ability to validate those antibodies in various applications in-house, such as IHC and the Western blot. And recently, we have been interested in how we can optimize and validate antibodies for use in multiplex applications to make it easier for our customers to get to their data faster.
So the goal is to get to a picture like this. So a 6-plex panel, how do we get there, and how do we get there cheaply with the least amount of effort? Because building the panel and actually validating the panel is what takes the most time. And so today I'm going to talk to you about two things. The first part of the talk is going to be really about how do you choose the right antibody and what are the validation practices that should go into choosing that correct antibody for multiplex. And then a simple panel building approach with our Pathoplex antibodies. So every multiplex system, whether it's the Codex or whether it's the Opal system or other multiplex system, really breaks down into 3 major workflow steps. And then you have to stain your sample, whether that is either at the benchtop or with a automated stainer. You have to image your tissue in slides, and then you have to perform image analysis. And most of the exciting, fun part is the image analysis and the imaging. However, you must get the sample staining correct, because if you have good sample staining and a good panel using validated antibodies, it makes the imaging and the imaging analysis much easier. In contrast, if you start out with a poor antibody, you're going to have background issues, off-target issues, high exposure, and all those things you'll try to compensate for with your imaging by adjusting exposure times, trying to get the best balance of signal, and you'll try to make image analysis adjustments, which complicates your workflow process. So it's best to start off this staining with a quality antibody, and you'll hear people in the multiplex field often state, we're going to start with a best-in-class antibody. What is a best-in-class IHC antibody? What does that actually mean? And what we hope it means is that it is an antibody that recognizes all of the antigen present in the correct tissue, the correct cell type within that tissue, and has the correct subcellular localization every time that we use that antibody. So it is sensitive and it's robust, and it is actually on target with only its target. Unfortunately, what best in class usually means is it's the best antibody you can find on the market that hopefully fits some of these criteria. And because you have your own time constraints, your own budget restraints, that you just have to go. At some point, you make the decision, this is the best antibody I can find, and you move on to your experiments. So how do you get to that choice of how do you choose an antibody? Commonly, what we hear from our customers is that they look to see which antibody is used in the clinic, whether it's a standard clinical diagnostic test or whether it is a companion diagnostic test. However, there are actually limitations to that. But the one thing that most people report to us and tell us how they pick a quality IHC antibody, it's the one that has the most most actual publications. And this is problematic in several ways. Oftentimes the actual publication record is not complete. Not everything about an experiment is reported in the literature. There are errors within the literature. And often you have a poor performing antibody that is propagated in the literature due to it being used first, or it was used in a high profile paper. So all of these things can either help you or hurt you. And I want to use actually PD-L1 as a case study of the danger in not vetting your antibodies in-house and making sure that you've chosen the best antibody. So over the past several years, there have been multiple research papers that have been published that are comparing different PD-L1 clones and their performance within IHC assays. Many of these clones are actually companion diagnostics. So these clones are being used to make a decision on whether a patient is included in a treatment plan or not. And so over the years, what we've come to find out is, as we've pulled these clones and tried to use them in actually multiplex or other IHC applications, that these clones for PD-L1 behave vastly different. For example, here's a non-small cell lung cancer tumor, and you can see that there's a range of staining quality between these 4 clones for PD-L1. And in fact, SP142 does not seem to recognize expression in the tumor cells very well at all. This has been confirmed by other papers. This is a paper out of the RIM group where they say the SP142 clone is an outlier that detects fewer tumor cells, and in fact, actually detects fewer immune cell expressing PD-L1. And then the other clones exhibit a range of staining in between. So all this has implications for patient outcome. But also it means that these clones are not interchangeable in your multiplex assay. So the lesson here is, so now we know about a PD-L1, that you have to be very careful. You just can't use what's in the literature all the time, because we're fortunate that the issues with PD-L1 is well documented. The problem lies with other antibody clones which have issues that are unknown to you as you're trying to set up your panel. So what are the strategies for choosing the correct IHC antibody? We kind of have a system in place that we do in-house, and of course that begins with gathering all the bioinformatic data that we can of the protein of interest, which includes expression data, and it's where it is actually predicted to be localized and it's observed localization. Because often the observed localization is not its complete localization, because only sometimes what's published in the literature is not the complete story. And we use sites such as Uniprot, Protein Atlas, and Pathology Outlines. And Pathology Outlines is a great place to get advice about clinical antibodies. We suggest that you use a search engine such as BenchSci or SciAb to let you know what antibodies are on the market, who are they sold by, what vendor sells the clone. And both BenchSci and SciAb now have images from the actual publications that can show you researchers using those antibodies. And if you don't have access to BenchSci or SciAb, just do a image search on Google. And it's a great way to look at all the companies or to look at all the images and examples of staining. And what you can see very quickly from that search is if you start seeing that the actual images show different pattern of staining, then that's a red flag that you're going to have to be extra careful purchasing an antibody for that clone. But then these last two options, these last two bullet points are what we believe are the strongest bullet point for choosing a IC antibody, and that is use vendors with quality validation procedures. They should be able to work with you. You should be able to call them up and ask questions, and they'll be able to help you. But when all that is said and done, the most important step is to validate any antibody in-house, right? And you should do this on known positive and known negative control tissue. Most of the immune markers, like CD3, CD8, etc., you can start on a tonsil and then move to your study samples. You can also source the tissue blocks that are known. And we highly advise that you bring in multiple antibodies to look at and to evaluate, and we'll show an example of why that's important. And we highly advise to use tissue microarrays. You want to know if the antibody behaves consistently across multiple tissue types. So how do researchers or vendors, how should you validate antibodies? The lowest bar of validation, and I would caution when using this type of validation, is cross-validation. For example, that's when you have a staining pattern for actually IHC and you say, is that staining pattern correct? Well, you look at a actual Western blot for that antibody and you say, oh, this antibody recognizes the correct band, so my IHC must also be correct. And I would caution you that with this, validation should be assay specific. So you should really not use one assay to validate another assay. You can use knockout validation. This is a extremely popular method and one that we sometimes employ here at Bethel, but it is limited to cell lines for humans. You really can't get knockout in tissue. Another common problem that arises is that the protein which causes the antibody to be off target may not be expressed in the cell line that you're studying. So you'll have a clean experiment. So the antibody does not recognize the protein of interest in the knockout cell line. But then when you move to tissue, it may be off target. So also there is confirming that there is actual messenger RNA expression in the tissue, or you can do mass spec analysis. The only problem with this method is that we all know there's a long history in the publication record where messenger RNA doesn't match up with protein expression all the time in every instance. We can use a method where we overexpress the target protein. And so when you compare the parent cell line to the overexpressed cell line, you should see increased signal in the OE line. One problem that we found with that is that using OE protein can cause you to actually select antibodies that see the OE, but does not see the endogenous protein. Perhaps the strongest way to validate an antibody is to use a intra-assay validation method using multiple antibodies to your target. And this is one of the recommendations by the International Working Group for Antibody Validation. And we believe in this very strongly here at Bethel. So by using antibodies to the protein, you should be able to get the same result, which gives you confidence. So intra-assay validation using multiple antibodies is, in our mind, one of the strongest ways to ensure proper validation of your antibody. And so how does that work? Well, you can design one or more antibodies to your protein from different areas of the protein. And in this case, we have 3 areas where we've designed a antibody to bind. And the ideal is that you test all 3 of these antibodies. And if these antibodies give you a similar result, it increases the confidence that your antibody is on target. And we started this program many years ago, working on polyclonal validation. And in this case, we use IP and the Western blot application to actually pressure test and actually validate our clones. And so we have 3 antibodies to 3 different parts of the protein, antibody A, B, and C. And we can use those 3 antibodies to IP the protein with, and then we can use those 3 antibodies to also blot those IPs. And when we see that the pattern of bands are the same, this gives us a high degree of confidence that our antibodies are on target. And so using multiple antibodies to different areas of the protein is a highly effective method to validate your antibody. And so we can apply this method to actually the IC application. Again, we are going to design antibodies to different parts of the protein, and we subject them to IC testing. And if the patterns of staining matches, we have a higher degree of confidence that the observed pattern that we see is the true pattern. Now, you notice I've not said anywhere that this proves that the pattern is true. All these methods are built around increasing your confidence. When you start saying that you've proven that this is the pattern, then it gets you in a trouble down the road. But we build in methods that provide us ever increasing confidence that the antibody is on target and recognizes the protein it should recognize. And so an example of this testing that we do here at Bethel for the IHC application is we use a tissue microarray in which we've put normal and tumor tissue cores, and we have multiple examples of each tissue type. And we will test multiple clones to see what their pattern of staining is. And this is an example that I have here is of a mismatch repair enzyme, MSH6. So we have 3 clones, and this is an example of a breast cancer tumor. Clone A and clone B and clone C exhibit the exact same pattern. So we're building that actual confidence that the pattern that we are seeing is the true pattern. We move to tonsil, clone A and clone B and clone C. Again, the same pattern, our confidence is building. However, when we move to ovarian cancer, we now see that clone B is exhibiting a different pattern from clone A and clone C. We move to colon cancer, clone B is still exhibiting somewhat of a different pattern. So in this example, clone B would fail a validation. And the important lesson is that a tissue microarray provides crucial information for antibody validation, because you do want your antibody to behave the same across different tissue types. And so in this example, clone A and clone C would be passing. And one important point I want to make about actually cross-validation, which is what we consider the weakest form of antibody validation. Clone B is identical to the other clones if you put it through a Western blot. So important to test and validate your antibody in the application that you're actually going to use the antibody in. And it's important to test multiple antibodies to ensure that you are making the correct judgment. So again, back to this screen, how do we choose the correct antibody? What we view as the most important thing is use vendors with quality validation procedures and also validate the antibody in-house. Right? What we also recommend is you try to use tissue microarrays if possible. We understand that is an expense that most people don't have in the budget. And so that's one of the things that we feel that we can do for you is we can validate our antibodies on tissue microarrays to give you more confidence actually in our antibodies. So we apply this validation strategy to our recombinant monoclonal antibody line. We call this line our Pathoplex line of antibodies, and we can apply each type of application screening. We can apply it early, and then we can confirm that validation so that we ensure that the antibodies that are coming out of our methodology and our platform are on target and recognize and function in the application that the way that you expect them to. So we're switching gears from how do you choose an antibody and the problems that can arise from antibody use to using these Pathoplex antibodies in a OPAL multiplex panel. So let's review just the general outline of the OPAL method. In the OPAL method, you bind a primary antibody to the tissue, and then you incubate that reaction with an HRP conjugated secondary antibody, and the HRP then will attach the OPAL to the tissue. You have a heat step, which removes the first primary antibody, and then you can allow you to come in with the second antibody. OPAL is attached to the tissue, and then you repeat the process for this third step. Now, one of the issues and one of the pain points that we hear from our customers that are performing multiplex technique is the time it takes to validate a panel and to choose the order of the primary antibodies. So for example, should the primary go in the first staining cycle versus the second or the third or the fourth or fifth staining cycle? So what are the ways that that the order impacts your multiplex success? So how does the order of staining impact your time to get a complete panel build, and how does it impact how your multiplex panel actually performs? So what we found as the most common problem is that the multiple heat-induced epitope retrieval events alter the antibody epitope antibody interaction in some way that we really don't quite understand. We believe it has something to do with the repeated cycles of actually heating and cooling. But this is the most common problem that we see, that the multiple heat treatments impact the ability of your primary antibody to recognize its target sequence. Another problem that can occur is that you can block the binding of an antibody. So there can be so much OPAL attached to the tissue that it blocks the primary antibody from binding in the next round of staining. Again, we don't see this often. The third thing that is also rare, but it does occur, is that you can trap your primary onto the tissue. And when you do this, this means you cannot remove that primary antibody with the heat treatment. And therefore, you can have crosstalk between channels. Now, again, this is a somewhat rare event, but we have seen it happen. So because of all these issues, you should test the order of the addition of your primaries within the opal staining. And this can be the most time-consuming part of trying to build a multiplex panel. And it costs in time, and it costs in the amount of tissue and the amount of opal and the amount of reagents that you're using. So we wanted to ask, can we come up with a way to build panels that's more simplified and can get you from panel design to staining your tissue the fastest way possible? And so we took 40 of our Pathoplex IC validated antibodies, and what we wanted to do is try to determine a staining order that we could provide to the customer, and that would allow them to quickly assemble panel options and then get to a completed panel faster. And so we chose to do this on equipment that we figured that we know most actual researchers have in their lab if they're doing any staining at all. And that is benchtop staining with a simplified way to do heat-induced epitope retrieval, just using a steamer, because we know from questioning, we knew from surveys that most people don't have access to other ways to antigen retrieve. And then we just used the we just purchased the opal fluorophore packs and the 1X amplification buffer from Akoya. So, and then the rest of our protocol you can find online. And so, using this simple setup, can we figure out a way to simplify panel design somewhat? And the way we did this is to test on 3 slides. So for a 6-plex assay, usually to test your antibody order of staining, you would use 6 slides in which you tested your antibody in each position. So first through 6th. So we're going to use fewer slides, 3 slides. In the first slide, we're going to stain at the first position and then go through each heat step all the way to the end. In slide 2, we're going to stain at the 3rd position. Again, do all the required heat steps. And slide 3, we're going to stain at the end and also performing all the heat steps. And so the goal of this is to see if we could use the information from this to figure out if a primary needed to be in the front of the staining order or at the end of the staining order, or if the staining order did not impact the performance of the antibody. And so here's some examples of that staining. So this is CD3. CD3 is an example of an antibody that likes to be in the front of the staining order. In contrast, FOXP3 is an antibody that prefers to be at the end of the staining order. And cytokeratin is an example of an antibody that does not care. It can function equally well first or last or anywhere in the or anywhere along the staining position. So what we did is we took that, built a table of staining orders, and then we wanted to test building a panel. So we chose actually CD68, cytokeratin, CD3, PD-L1, FOXP3, and CD8 to build our panel. And excuse me, I got ahead of myself there. What we found in our results was that 4 of our antibodies like to be at the front of the order, and most of them like to be at the back, but a number of them could be in any position. Okay, so back to the panel building. The antibodies that are in tan are what we're going to choose to build our panel. Right, so we just simply fill out we use the table to fill out our order. So CD3 likes to be up front, cytokeratin and anti-CD8 can be at any position, front or back, and actually PD-L1 and FOXP3 likes to be at the end. So we can start filling out our 6-plex panel. So we'll put CD3 in the first position. We'll come and fill in with cytokeratin and CD8. And we have CD68 that likes to be 3rd through 6th. So we'll put it in the 4th position. But then we have a problem because both FOXP3 and the PD-L1 antibody like to be in the 6th position. So that gives us 2 options. We can have option 1, where we put the order with FOXP3 at the end, or option 2, where we put actually PD-L1 at the end. So the idea is that we then proceed to test which option is the best. Again, we test on we always test on some type of array. This is a tonsil array. Again, we're starting with tonsil because tonsil allows us to see where to actually check to see that our staining is giving us the results we should. For example, we can see the T cells. We can see the CD8 is a subset of CD3 as expected. We can see that we have correct expression of the other markers. So this is a zoomed in view. This is 20x. So up top is an example of all the colors, and in the body images, we pulled out the FOXP3 signal and the PD-L1 signal. And we can very clearly see without doing any type of image analysis that option 1 seems to be the best option. However, if we do inform image analysis, we see that the 2 options for CD3, CD8, CD68, and cytokeratin are the same. Of course, they're in the same actual position. But there is a huge difference between option 1 and option 2 for FOXP3 and actually PD-L1. So you're detecting fewer cells. So the order of these antibodies does impact your eventual image analysis. So now we move from tonsil to a test tissue. So this is non-small cell lung cancer. Again, we have our panel. You zoom in. And again, option 1 appears to be the better option. This time it's not affecting FOXP3. It doesn't appear to affect FOXP3 as much as it does actually PD-L1. But again, we can do our image analysis and we can confirm that there is a large difference between option 1 and option 2. So what we have is we have a way to provide our customers with a simplified way to build a panel. And that is, you simply read the staining order off of our tables, and you use those to build a options of panels. You're going to have options of where to put them. Hopefully, we reduce that to the fewest options available. And then you're going to assign an opal. And really the large recommendation that we have is that you separate targets by at least 1 opal color, that if they exist in the same subcellular compartment. We used to recommend that you avoid the far red fluorophores for low abundance targets, but the opal 690 and the opal 780 perform extremely well. And most of the time, this is not an issue. I mean, FOXP3 is not a very abundant protein in most tissues, but we can see it easily with the opal 780. So then you would just test these initial staining orders, pick your finalized order, and then if you need to, you can balance the signal by actually adjusting the opal concentration at the end. So we believe that this system will help you get to your panel faster. So we hope it will decrease the time from design to actually staining your tissue, to actually getting the answers in your experiment. And so one of the future directions that we want to do, we started with on the bench staining because that's what most of our customers can do. We also wanted to transfer the Pathology panels to a auto staining platform, and we've chosen the BioCare medical platform, which is a nice platform because one of the applications it's designed for is actually multiplex staining. And we've been able to verify and transfer for 3 of our panels over to the staining platform. So we're very excited about this, so that this simplified method of panel building works on the bench top, and then we can easily transfer that to a auto stainer. So I'd like to thank lots of people at Bethel, but primarily our IHC group who did most of this work, and of course the protein expression group and the monoclonal antibody cloning group, which actually produce the antibodies that then we are that we have the pleasure to test for IHC applications. So with that, I will end my talk, and thank you very much.
Thank you so much for that presentation. I hope our audience found it of great interest and value. If you do have any questions for Mike, then please do continue to submit them using the Q&A tool in the right-hand side of your screen. So far, we've received a number of questions, so we're going to address some of these now. So first up, Mike, do I need to validate every antibody I buy?
The short answer is yes. The level of validation will be determined by your own needs, but as a baseline or a actual minimum of testing, you need to test the antibody on samples that you know to be positive to determine that the antibody functions across the recommended dilution range on those known positive tissues, because without that information, you can't proceed with optimizing your panel.
Great, thank you, Mike. So next question, what are the advantages of using an amplified system such as Opal?
That's a great question, and the biggest advantage is an increase in your signal to noise ratio. So the amplification by Opal or other pteramide systems really allow you to boost your signal up to a usable level. That's the first advantage. The second advantage is it allows you to use antibodies for the same host species. It can become quite hard to find antibodies in the traditional multiplexing methods that you would need to mix a rabbit antibody with a mouse antibody with a rat or other species antibody. The pteramide or Opal systems allow you to use antibodies from the same host species. And finally, the biggest advantage is if you already perform any type of IHC in your own lab, you can learn to do the Opal system to actually bring the multiplexing into your lab.
Thank you, Mike, for that brilliant insight. So our next question that's come in, why can I not just begin staining my samples to work out conditions for a multiplex panel?
Well, this really goes back to the first question, is should you validate your antibody? Yes. And in the case of when you're beginning to work on a multiplex panel, you again must confirm that your antibody behaves in the correct way. And this has to be done in a tissue that you know is positive for the protein that you're trying to detect. So the best case, the best results are when you work with tissues that you know express the protein. You can optimize your antibody, because sometimes your samples may or may not have high levels of expression. So when you're validating your panel, you want to begin with tissues that you know have expression. The second issue is that your sample is actually precious. and you don't want to waste your sample on the optimization process, which can use a lot of tissue and take up quite a bit of time. So the best case scenario is start on tissue that you know expresses your protein of interest, and then once you have the first optimization steps worked out, then move to your sample tissue.
So our next question is, if an antibody works in one multiplex system, can I just assume it will work in a different multiplex system?
In general, no. But let me back up and say that if you see that one antibody is working in one platform, it certainly is actually predictive that it could work in some other platform. But remember, one of the things is that when you're using an amplified system like the Opal system, and you move to a multiplex platform that doesn't have that type of application, then you may have signal to noise. You may have signal to noise issues that become actually problematic. So again, when you change platforms, you will want to re-optimize your antibody and actually validate that the antibody works for that particular platform.
Thank you. So an interesting question here. Should I trust the data on a vendor's website?
This is an interesting question, and it is not an easy one, but in general, I would say you should trust but verify. So I don't think that vendors are trying to put out information that is wrong, but you should be able to take their data from their website or from their from the information they provide you, and you should be able to replicate that in your own lab. Now, there are some red flags to look for. If you look at the if you look at the data on their website and you start to see, data that doesn't agree with what is in the literature, then that might be a red flag. But in general, I think all the vendors are working very hard to put the best data that they have forward, but that doesn't mean that you should not verify that data in-house. And vendors should be willing to work with you to actually verify that data and to help you along with those experiments.